Good morning, good afternoon, good evening. I don't know wherever you are. Finally, welcome to this webinar. I am Stephane Simon. I am a professor in endodontics and oral biology, and I used to be a professor at the University of Paris, and then I quit it a few years ago. And now I'm back to my private practice, and I'm still a teacher and doing some clinical research on very specific projects. So in this webinar of 20-25 minutes, I'm going to share with you some results we recently provided for a clinical research to compare the BioRoot and the BioRoot Flow and to see if there is any difference in terms of outcome. But behind this research, which was a comparative analysis, but behind this, I was definitely also interested to see if using a bioceramics with a single cone technique is a predictable and reliable technique in every hands. So before to start this presentation, let me tell you that I do not have any conflict of interest regarding this product. So I don't have any money back for any feedback money regarding the sale of the products. But I have also to declare that I am the principal investigator of this clinical research. I say I am and not I was because this research is still ongoing, because after 2 years recall, the results were so interesting that Septodon has decided to continue to pursue this research with a 5 years recall. So that's why we are still doing some recall, and today I'm going to present you the 2 years recall results. So regarding the filling and the obturation, I just come back to these ESE S3 guidelines. I'm pretty sure you know them. If you don't know them, just go and Google and ask for the PDF, which is free for download. And it's very interesting because the previous guidelines were published in 1996, and though the ESE has decided to provide new guidelines on different topics. The objective was to provide some guidelines based on the literature. And if you read these guidelines in every topic, instrumentation, irrigation, whatever, you see that the research behind this is quite poor. And finally, as a scientific organization, it's very, very difficult for them to provide some guidelines. But what is interesting in the guidelines regarding obturation is that you can see that the root canal filling with good aperture in combination with any of the included sealers, epoxy resin, Zoe, and calcium silicate. That's the first time we can see the calcium silicate coming in some guidelines. So they don't say how to use it as a sealer, to seal the cone behind themselves, or to use it as a filler. I just pour it into the root canal, you fill it, and then you get a single cone. So, but one of these, actually, they say that it's in the guideline, you can use a calcium silicate. So that was a good point. All the techniques we use today to fill a root canal, all of them are based on using good aperture as a filler and a sealer, whatever it is, zinc oxide or epoxy resin. This sealer is here to seal the cone, to make a sealing between the good aperture point and the dentinal wall, because the good aperture, it doesn't stick on the wall, and then we need a sealer to do the connection between both, and we know that the sealer is a very weakening point. So the only thing which is going to be infected in the root canal, that's the sealer, and that's why we need a maximum of good aperture and a minimum of sealer, thickness of sealer. And that was a single cone with a zinc oxide sealer is not reliable, because there is too much sealer, which is not a filler, and that's very important, these two words, filler and sealer. So the sealer is dedicated to disappear, and then the more sealer you've got, the highest risk of reinfection you have in the root canal. So that's why the root canal filling based on good aperture, you have to get a maximum of good aperture and a minimum of sealer. So what are they talking about? So you can see here that with whatever the anatomy of the tooth is, that you can see here on this premolar, it's a very complicated anatomy. So not complicated, but not the normal anatomy. And it's a young patient. And then when you fill the root canal after the disinfection, and you maintain the disinfection ongoing so the apical lesion disappear. We know that. We know that the problem is an infection problem, and then the consequence is inflammatory, and then removing the infection, you get the healing. And what is interesting is that even if your technique is not very accurate, so even if you don't reach, I would prefer to do that, even if you don't reach the final goal, meaning taper preparation, very, very nice obturation, if you disinfect it and you maintain this, you get a healing. So I completed this case. I'm not very proud of it, but I was not able to find the patency of the canal, of the major longus canal. But whatever I used a lot of stuff for disinfection, and finally I was able to get a healing of the apical lesion. The game changer of endodontics, that the evolution of endodontics is moving to a less invasive techniques. We know that if you want to use a warm compaction, you have to get a nice taper, minimum 6%. And then we remove a lot of dentin just to be able to fill the root canal with this technique. And somewhere, it doesn't make sense to say, okay, I'm going to fragilize my tooth only because I want to use this technique of root canal filling. If you do a retreatment, the question is completely different. Why? Because the tooth is already enlarged. The canal is already enlarged. So you don't fragilize too much the tooth if you retreat the tooth. But for an initial root canal treatment, where the canal is free of everything, doesn't make sense to remove dentin just for fitting with the recommendation of the root canal filling technique. So that's what we have to talk about. Because the evolution technique, look at this guy, Steve Buchanan, who everybody knows him. And I remember when I was a young dentist, he was pushing the GT5. The GT5 was a 12% taper. And actually, Steve Buchanan is selling some the mini cuts which are 3% taper at the tip. So what is interesting is that finally, even the people who were Schindlerians in the brain are moving to this less invasive with less preparation of the root canal filling, of the root canal. And there is something which, so of course it makes sense, but there are some limitations. Can we disinfect the root canal, and can we fill it? So I'm not going to talk about the disinfection, but these disinfection techniques must be revisited, and the filling techniques as well. And that's why the bioceramic is interesting, because it can fit with a less than 6% taper preparation. So the concept of using a bioceramic filling is to consider the bioceramics or the hydraulic cements, whatever the word you want to call them, as a filling material. You are going to fill the root canal to obturate it with something you are going to inject inside. It's not a sealer. Of course, it's a sealer somewhere, but it has not to be used as a sealer. And I want to be very clear with this. And that's why the BioRoot RCS, root canal sealer, for me, it's a mistake. Because it's not a sealer. It would have been BioRoot RCF, root canal filler. The good apical point we're going to use must be considered as a carrier to push the material in the apical third, and eventually to retreat the tooth if we have to do a retreatment or if we want to place a post. Because the bioceramics would be very hard, so you won't be able to go through if you have to remove it for the retreatment of the tooth. But also, if you want to place a post, you have to remove at least 1/3 or half of the root canal filling material, and then you need the good aperture just for that. So finally, the technique is a single cone technique, but the philosophy behind this has nothing to say. I'm filling with the material, and I'm using a good aperture point just for the techniques. If I want to fill a root canal only with the bioceramics, there is no problem. So what are the questions? So for me, it's an evidence because I'm using this for 12 years now. And the question is that, is this technique reliable and predictable? The second question is that, do we have clinical evidence of this effectiveness of this technique? Do the products influence the outcome? So we have many products on the market. It's that, all the bioceramics provide the same results? And then though the carrier influence, the carrier is not a good applicable. So the carrier is something we add to, the company add to the material to make it like a flowable, putty, you can inject it, or sometimes like the BioRoot, you have to mix powder and liquid. So now with the BioRoot Flow, you are going to inject the material inside the root canal. So you need to add some carrier to make it like a creamy. And is it science or purely marketing? That's a big question, because finally, like everything, everybody remember the resilient problem. So where the marketing comes through, and they say that's the best technique. A lot of speakers giving lectures all over the world, and then 2 years later, the results were catastrophic. So we still have this in our mind, and that's why a lot of people are very they are not very happy to see something new, different, something new coming, and that's why the bioceramic story is a little bit complicated for a lot of people. So the state of the clinical science, when you look about the papers published, finally, it's not only for the bioceramic, but in average in endodontics, but this slide is focused on the bioceramics and the BioRoot. When you look about the outcome, there are more case series, very small, very few retrospective study, very few prospective study. So the power of the science behind this is not very, very high. And for me, not sufficient to say, okay, there is a scientific evidence that this product and technique is good. Actually, when you look about what are the experimentation going on, very few of them. So you can go on the website, and some of them are comparing AH plus BioRoot or MTF Eliplex, AH plus Endocil and the sequence, etc., etc. But a lot of very few, not a lot, but very few experimentation are still ongoing for recruitment. But now, 2 years later, they are still recruiting, and I'm not sure that they will go further. So what was the problem of the BioRoot RCS, except in the name of RCS? What was the problem? The limitation was to use a powder and liquid, and sorry, it doesn't work. So we had to mix the powder and the liquid, and finally this mixing was first it was not very the nurses were not very happy. All of you remember the 5 drops, big drops, small drops. So the mix of the products was not always the best one and the more creamy one. So sometimes it was too liquid, sometimes it was too hard. So there was something very complicated to manipulate this product. So that's why Septodon decided to provide a ready-to-be-used product. So the BioRoot Flow is exactly the same products on the paper than BioRoot, but they add a carrier to make it creamy and ready to be used as an injectable material inside the root canal. So for me, it will be complete. So sometimes I don't call it BioRoot Flow because I've called him the PA 1704 for 3 years during the experimentation. So I have to be very careful. So the BioRoot Flow is a commercial name of this product. It's exactly the same than the BioRoot, but you don't have to mix it. It's a syringe, and you are going to inject it inside the root canal directly, and also always add a single cone of gutta perca. So what is this experimentation I'm talking about? Initially, the objective was to collect clinical performance and safety on the use of BioRoot Flow, because this product is ready to be launched on the market for years, and it was launched in the United States more than 16 years or 18 months, sorry, not years, months, sorry. And it was not available in Europe. Why? Because it's an injectable, it's a class 3 medical product, and then as a clinical 3, the regulation ask for clinical experimentation with a randomized clinical, and then they ask the company to provide results that first the problem is safe, the product is safe, and secondly that it's a good product, at least not as good as the one already on the market. So the sponsor was Septodon, and still is. The CRO was SLB Pharma in Rennes, a very nice team. If you want to do some clinical research, I will recommend them because they are very, very cool, very nice team, very accurate. So congratulations to them. And I was the principal investigator coordinating a team of 13 investigators on 7 sites, so in France and Belgium. But what's interesting is that we mix endodontists and general practitioners as well. So all the treatment were not completed only by endodontists. And for me, it's very important. Why? Because most of the endodontists, they are very happy and very accurate with the worm compaction. So finally, I think that in the hand of an endodontist, everything works. So what I was interested in that to see that if with the GP, which is not very happy with endodontist but want to do the best as he can, is this material working well? And having this mixed team we will be able in the future to compare the results of the endodontist and the results of the GPs. Of course, everything is not based on the material. I'm aware about that, like you. But if at the end we have similar results between endodontist and GP, that asks a very nice question. So we didn't ask the GPs to do the endodontics in a very specific manner. So we just asked them to use the rubber dam in every cases to be able to not to include some very calcified canal with complicated anatomy, and that's it. So they can use the files they want, they can use the irrigation technique they want, like the endodontists, some use reciprocating, others use rotary files, and finally, I didn't care about that. The only thing is that you have to use a rubber dam. You can use a microscope or not. If you don't have any microscope, it was not a requirement for this experimentation. So here are the name of the people who were in charge of coordinating the team on site, and then we had 13 investigators. So the study design was that we included 160 patients. So the alpha risk was calculated before, and then because it was randomized we wanted to randomize the cases between BioRoot and BioRoot Flow. And then we had 160 patients, and then the system was settled for having 80 patients for the BioRoot Flow and 80 patients for the BioRoot RCS. And at the end, we had we completed the treatment. Visit 1, visit 2, we checked the coronal restoration and the visit. We called the patient at 1 week, and that's to see that if they had pain, and then we evaluated the pain, the discomfort, if they had to take pills, etc., etc. After 6 months, we had a first recall, and so just because we wanted to see the evolution, but also we wanted to be sure that the tooth was being restored with the coronal restoration. And after that, we had a visit 4 at 12 months, visit 5 at 2 years recall, and now we have the visit 6 at 3.5 years, and the visit 7 which will be at 5 years, and it's going to close the experimentation. What is important is that visit 6 and visit 7 are only ongoing for patients who receive the BioRoot Flow. We are not recording the patient who received the treatment of the BioRoot RCS. So there are a lot of inclusion criteria and exclusion criteria, like every clinical research. I'm not going too deep inside, but we just asked the GPs at least to exclude the root canal treatment on tooth with a calcified canal and with a suspected perforation, because we didn't want to modify the outcome just because the preoperative conditions. So the chart flow, you can see that patients were allocated, and then we started the first inclusion on the 6th of November 2020, just after the COVID lockdown. And we completed the last inclusion of 160 patients on the 26th of June. And what is interesting that we had a very, very nice follow-up rate, because at 24, 85% of the patients had been recalled. So after that, we had plenty of data that we're still analyzing. So we had 50% of male and female. We had we made a difference between root canal treatment and retreatment, teeth with apical lesion or not, and the tooth types. So we had a majority of molars and premolars and very few incisors. So for the results, we provided to 2 calibrated endodontists with a kappa coin at 0.79. I'm not going to explain this, but if you're interested, go in the literature and you will see how we provided. So we had 2 readers calibrated, and then we asked them for a first lecture. When they had disagreement, we asked them to do a second lecture of the X-ray where there was a disagreement, and then at the end, if finally they didn't merge, and then they still had the disagreement, we had a meeting together and we discussed the case for final rating. So for the 24 months results, we selected the loose criteria and the strict criteria. Like every research in endodontics, actually, the loose criteria were successful treatments plus uncertain. What we call uncertain, that was, for example, the apical lesion in the process of healing, and where the lesion didn't evaluate there through the healing, but didn't evaluate to the pathology. And the strict criteria were strict successful treatments only, and then that allowed us to evaluate the efficiency of the treatment. So safety analysis, we had only one SAE, but it was an SAE for pulmonary embolism, so it has nothing to see with the products. And so finally, all this data will be published. We also were interested in the postoperative pain. Do they have less or more postoperative pain with one product compared to the other one? And finally, there were very few difference, and the BioRoot Flow was a little bit lower. We had a lower number of postoperative pain, but it was not statistically significant. So very quickly, I give you this presentation, this data, because if you want all the data, I can speak about that for 3 years, but we don't have the time, especially for a webinar. So the success rate were 91% or strict criteria 89%. So that's why you will have a lot of communication from Septodont telling that they have 91% of success. So the 91% is not the loose criteria here. That's the average of all the treatments, including retrainment, treatment, etc., etc. So here we are talking only on initial treatment. 91 compared to 89%. Remember, they are endodontists, but also GPs, and some of them were very young. So if you look about the literature, there are a lot of papers showing that a huge difference between the endodontists versus GPs. We didn't have such a difference in this experiment, and that's why we merged all the data. For the retreatment, it was close the same, a little bit less with 82.5, with the strict criteria compared to the 89 to the previous one, but that's retreatment. We know that the success rate of the retreatment is a little bit lower, but with the loose criteria, we have a 90% instead of 91 with the initial treatment. The teeth with apical lesion at the baseline, so we see that we have a 95 and 96. So this was surprising because it's a little bit lower, higher than the previous results. But just because we had we had less teeth with apical lesion, so the number of cases is lower. So if you have healing, you modify the success rate and the percentage of success just because you have lower cases. So it doesn't mean that's why we have to be very careful in comparing the results from one part to another one. And teeth without apical lesion, the baseline is a little bit lower. Why? Because we had more treatment, we had more teeth without apical lesion compared to the teeth with apical lesion. So the discussion is, okay, at the end, I remember my mentor told me many years ago, he said, Stephane, we do not discuss results. The results are here. We can discuss the way we completed the research, if you want. I know how we did it. I know that it was very strict, and definitely it was a good experimentation. It's the first randomized clinical trial done with the bioceramics fillers. And for me, I'm very happy, okay, for the material, that's no discussion. But also I'm very happy that finally we have some data showing that a single coat technique with a bioceramic filler seems to be a reliable technique. So after that we can discuss everything. Okay, it's only 160 cases, but how many research are done on 160 cases in endodontics in a randomized way? Very few of them. So that's interesting with this material because remember it's a class 3, and on my knowledge it's the only one classified as a class 3 material. And then even if you have some extrusion of the material like puff or big puff, it's not a problem because it has been tested and it's an implantable material. And as you know, it will disappear because the osteoclasts, they love silicate of calcium silicate. So of course, do not fill all the lesion with the calcium silicate, but as a class 3 material, if you have an overfilling or an over-extrusion, it's not a big matter. So, as I said to you in the beginning of this webinar, we have 3 more years of follow-up coming on. So we have we are completing the 3.5 years follow-up, and then we'll have the 5 years at the end of 2026. And to complete the results, it will be interesting. So my conclusion is that finally, at least me, I can consider that filling root canal with the BioRoot is a relevant technique. So it is the only randomized clinical trial completed on the evaluation of efficiency of this kind of material, and we can consider, or we have one step to say, okay, it's a reliable and predictable technique. Doesn't mean that it's the best technique and then we have to stop the work compaction. Absolutely not. If you can do and you are happy with the warm compaction, it doesn't make any sense to change your technique. But don't use this material as a sealer. It doesn't make sense. Because with the warm compaction, you need an oily material. So you need a pulp canal sealer or whatever, something done for that. And the bioceramic, there's a lot of discussion. Can you warm it or not? Okay, I think it's not a problem to warm it, but these products are not made for that. So why do you want to use a material not dedicated for this technique just to say I'm using bioceramic? If you want to use bioceramics, do them as a filler. If you want to keep warm compaction, vertical compaction, and I'm still keeping this for such cases, finally, do it with appropriate sealing. So we must keep in mind that filling is efficient as long as the previous stages of treatment were completed in the state of the art or objective. Okay. Of course, if you do not shape and then either disinfected the root canals, so the filling is not going to provide good results. But it's not because of the material, it's just because the treatment is not completed in the right way. So if you have already seen me in different occasions in the meeting, you know that I love this sentence of the three steps of innovation of Schopenhauer. So the first step is a new concept is always ridiculous. The second step is the concept is the aim of a strong fight. Actually, with the bioceramic, we're on the second step. And the third step of everything, it becomes an evidence. And hopefully it could become an evidence in a few years now. And so I hope I will see that, because I'm very confident in this kind of products. So can we consider the bioceramic filling as reliable and predictable? Based on this result, I would say yes. Do I still use a warm vertical compaction in my practice? Yes. If yes, when? Mostly for complicated retreatments, when I'm not able to clean completely the root canal surface, because the bioceramic has to be attached to the dentin. So if I see that I have some places of sealer that I cannot remove, and I'm pretty sure you understand what I mean. In that case, I'm going to use a warm compaction because I need something to come and fail, and then the bioceramic is not going to stick on the sealer or remain in sealant. Do I recommend to switch your filling technique? Absolutely not. Absolutely not. I'm not here to say what you have to do. I'm just here to share my experience and my point of view and do just whatever you want with this. And probably you will need more lecture, more evidence if you want to move. And again, if you use a warm compaction, it's perfect. Don't switch to anything. The only thing is that if you use a cone sealer, a single cone with a sealer, I will encourage you to do something and to change your technique, because that's not a good technique. But if you use a thermocompaction, if you use a thermofill, if you use warm compaction, even if you use lateral cold condensation, that's good. These techniques provide good results. Are they as easy as a bioceramic filling? I'm not sure. It's you make your decision. But the aim of this webinar was to show you that now we have clinical evidence that if you do this, it works. Thank you very much for your attention, and see you soon.